Thank you for joining us for Psomagen's Introduction to 10x Genomics Single Cell Flex V2, also known as Apex assay. Before we get into how the Flex is new and improved and different, I wanted to go back and look at just a standard gel emulsion at the technology itself. And the real magic is in those gel emulsion beads that you see there, the 10x barcoded beads, and also combining that with oil droplets so that you get a single cell and a single gel bead into the same emulsion. And then you can have that unique barcode applied to all the transcripts in that emulsion and a unique molecular identifier, which is also great to rule out multiplex. Okay, the magic here is in those gel beads, and also the price is in the gel beads. Now, the Flex assay changes the order of things, and the benefits of this are quite dramatic. Each sample is first hybridized to a whole transcriptome probe panel. And this is human or mouse. It's probe-based because it's for fixed RNA. Next, each individual sample can get a unique 10x barcode added to it. So now you have a unique barcode on each sample. You can pull those samples together and just do one round of gem creation instead of doing 4 separate ones like you would have had to do in the past. The probes get ligated inside of the gems. Everybody gets translated to reverse transcribed to cDNA, and then you can partition those pools on the chip. It can hold up to 1 million cells or nuclei per each lane of the 8-lane chip, which means you could theoretically do 8 million cells or nuclei all at once. And then depending on how many you have, you can end up with just one single gene expression library for all 4 of these samples and save on the sequencing cost too. And 10x was nice enough to create this experiment builder inside of their store. So you just go in, you pick Flex, and you say continue. And I want to point out that it works really well for small studies. Once you get over like 16 samples or 320,000 cells, it starts to get confused. So you're better off reaching out to your FAS or science and technology advisor. In this example, I picked human, 12 samples, 20,000, and its nuclei, since we get a lot of fixed tissue requests. I think it's our most common sample. And then it will kindly show you that you barcode all the individual samples, you pull them together for gem creation, and then you only run 1 lane of the chip for the partitioning step, and you only have 1 library to sequence. So you've just saved buckets. My favorite part about this is that in the 96-well oligo plate, each well can label 20,000 cells. And so this isn't a one-time use and throw it away. You can have 5 samples, you can have 12, and you can keep coming back to that plate until you've used all of the samples, all of the oligo barcodes in there. It's a huge saving for customers, and it really is a great enabler of longitudinal studies, archival samples, just being able to expand researchers' budgets to cover more and more cells and more science. Thank you very much.